going to be looking at metabolism and trying to understand how chemical reactions are used in a cell to provide energy for that cell. So there are some eight elementary statements, guidelines for metabolic processes within cells that first off, every cell actually acquires nutrients. Um, and metabolism requires energy to do something with these nutrients. This energy can come from light or the catabolism of other nutrients. Energy is stored in adenosine triphosphate, ATP, and that is the energy storage molecule that's used for just about everything in a cell. Cells will catabolize nutrients to form precursor metabolites. And when I'm talking about precursor metabolites, I'm talking about the little monomers, the little molecules that are going to get put into bigger things through anabolic reactions that will use ATP and other enzymes to create these larger molecules. Sometimes they will be big enough to be called macromolecules. And so enzymes plus ATP will help to form these precursor metabolites into macromolecules. And it's by these macromolecules that cells grow by assembling them together, and cells will reproduce once they have doubled in size. So we're going to look at catabolism and anabolism. Catabolic pathways are going to break larger molecules into smaller products. Taking big molecules and breaking them down, anytime you break a bond, it's going to release energy. So these are exergonic reactions. This released energy will get stored into some other molecule, maybe into ATP, but always any reaction that takes place, some of it will be lost as heat. And so the smaller products will be used as precursor metabolites later for anabolism. In anabolism, we will then take these small molecules and synthesize them into larger molecules, and this will require energy. It will be an endergonic reaction because energy that has been released before will be used here, brought into this set of reactions to put together bonds. So you store energy into the bonds. And of course, some of that will be lost as heat as well. And so we see here that you have larger nutrients brought into the cell. Through catabolism, they will be broken down into precursor metabolites. The energy that's given off through the breaking of bonds will be stored into ATP. Some of it will be lost as heat. We can't help that. Those precursor metabolites will then be used to make things that the cell needs, like larger building blocks or macromolecules. Along the way, to do that, ATP will be used. They will be broken down to give off energy, and that energy will then be used to make these precursor metabolites into larger building blocks, will be then used to make larger macromolecules, which will turn into either cellular structures like membranes and ribosomes, or be used in cellular processes like cell growth and cell division, or in energy storage for later use as carbohydrates, which will then can be broken down through catabolism, giving off energy, and as you see this cycle work its way all the way around. Now, as anabolism and catabolism are paired, if you do catabolism, you're going to end up doing anabolism afterwards, and then you will do catabolism. We see this also with redox reactions, oxidation reduction reactions. And some of what we're seeing with anabolism and catabolism is going to be using redox reactions, where we will be transferring electrons from an electron donor to an electron acceptor, where reactions will also are going to occur simultaneously. The handoff of an electron is going to be the receiving of an electron by the other molecule. So a molecule is reduced, a reduction reaction, when it gains an electron, or it gains an electron that is part of a hydrogen atom. A molecule is oxidized when it loses an electron or a hydrogen atom, or it gains an oxygen. We're going to be focusing on the electrons though, and note that Leo the lion says GER. Loss of electrons is oxidation. Leo says GER. Gain of electrons is reduction. So please make sure that you do keep this in mind. I wished I had learned this when I was maybe a freshman or sophomore in college. Unfortunately, I didn't learn Leo says GER until I was in a senior, and I didn't. Wished I had known that little phrase that would have made things a lot simpler on me in my chemistry classes. So when you give an electron off and it's accepted, that's oxidation. The reduction will be when that, um, so let me say that again. You lose the electron, it's oxidation. And when that electron is then gained by something, it is reduced. Of course, that makes sense because you're reducing the charge, adding a negative um, part of a part to this molecule is going to reduce its charge, and so that's why it's a reduction. So what we're going to see is that we have these molecules that are called electron carriers that are used in redox reactions. And so cells will use electron carriers to help carry electrons. They funnel electrons back and forth. A lot of times they'll use hydrogen atoms as the whole molecule, the electron with the hydrogen attached. And so there are three important electron carriers that act like taxis, moving electrons back and forth as needed. NADP plus, NADP plus, and FAD. Those are the non-electron holding versions of these. When they accept the electron, they become NADH or NADPH or FADH2. And so what they'll do is they'll go drop off the electron and they get turned back into NAD plus or NADP plus or FAD, and they'll pick up electrons and then go drop them off again. They go pick up the electrons and go drop them off again. It's kind of like a taxi service. Now ATP production is also necessary because it's the molecule that holds the energy that's really important for energy storage. So organisms will release energy from nutrients through catabolism, and then we can take that energy and store it into the bonds of ATP. So adding on another phosphate to an ATP molecule is actually going to be really helpful in holding on to energy. This is called phosphorylation, where we take an inorganic phosphate and we add it to our substrate. And so basically we see something like this. A D, and D stands for diphosphate, P, plus Pi yields ATP. Now, this ATP can also have that phosphate taken back off, and we can reverse the reaction where we take ATP, take off the phosphate, release energy to be used through anabolism, and produce ADP again. And then, of course, we can go backwards again. So we see this reaction being reversible. So cells can take ADP and turn it to ATP in three ways. One is substrate level phosphorylation, where the phosphate is transferred from something else that has a phosphate, another phosphorylate compound. Or you can do it through oxidative phosphorylation through a redox reaction, where energy, such as in the respiration equations that we see, the respiration reactions, are used to take inorganic phosphate and attach. And this is what we've described here so far. This is what we're going to be working with probably the most. But there's also the potential for photophosphorylation, where light energy is used to take inorganic phosphate and attach it to these molecules. Okay, and so anabolic pathways will use some of that energy that ATP is holding on to, and by breaking off a phosphate bond and releasing some of that energy, and we'll pass it on to whatever the anabolic reaction is trying to make. Now, in reactions, there's also these things called enzymes, and they are organic catalysts. They will help to increase the likelihood that a reaction will occur. They do not make a reaction happen. The reaction has to be able to happen on its own, but it might not happen fast enough in a cell or in a body to keep that cell alive. And because of that, we see enzymes used throughout living things to help speed up reactions so that they happen faster or more likely to happen, to make sure that biological living organisms stay alive. Now, what an enzyme does is to reduce the activation energy necessary for a reaction. You always have to add a little bit of energy into a reaction to make it happen. It takes a little bit of work to make something, occur. And in this case, that's what this hump is. Now, without an enzyme, the hump is rather large. It takes more energy to make the reaction happen, so the reactions happen more slowly because it takes time for that energy to build. Enzymes, on the other hand, will reduce that activation energy to this point where the reaction can happen a little easier and will happen more fast because it doesn't require as much energy to happen. We have to always have some little bit of activation energy, otherwise the reaction would just happen from bang to bang. It would happen from here to here, and we would just be having reactions happen all over the place even when we don't want them to. So this is why we don't just spontaneously combust all the time. We don't have enough energy into for that to occur. It's because it requires some input of energy, which is why paper doesn't just start to catch on fire on its own. You have to add some fire to it to create that energy that's necessary to get the reaction started. Enzymes, on the other hand, are kind of like adding maybe some kind of like gasoline or something to it, so it makes it easier for the fire to start and burn. So it reduces the amount of wait time and energy needed to make this happen. And of course, this kind of reaction here, as you see, this is where the reaction has more energy and the product has less, as we see it go down. And so this is a release of energy. Enzymes work this way, where there's an enzyme grabbing the substrate in its active site here, and it will put pressure on the bonds of this molecule so that eventually it can break that molecule into pieces, releasing energy, and then the enzyme will release the substrate as the products, and that enzyme will continue to be able to do it again over and over and over. It's a reusable thing. Now there are some things that can affect the rate of reactions that are enzymes. Temperature. One, if you heat up a reaction, it works better. So up to a point. There's a point at which you heat it too much and then the reaction stops working, and that is called denaturation. You can also change it by pH. There are certain pHs within our bodies, like for example in humans, there are certain enzymes in our stomach that work better at low pH. And you have a hump here over at low pH, which is acidic. And then other parts of our body where it's going to be more close to 7, and that's where we see the hump, where it works best. Really depends on the enzyme at where the best pH is going to be. Once you're outside of that pH range, more acidic or more basic, you get less functionality of that enzyme. You also can affect how fast it works by adding more enzyme or more substrate concentration, but that only helps up to a point, and then it doesn't really help anymore and it levels out. And then there's also the option to induce inhibitors, where inhibitors will actually affect the binding of an enzyme to a substrate. They won't be able to come together. If they can't come together, then the enzyme cannot do its job. So if you don't form that enzyme substrate complex where they're actually attached, enzyme in the active site, then you won't be able to get that reaction happening as fast. The enzyme is not being helpful. Now, I want to talk about some specific reactions, in this case the carbohydrate catabolism, so the breakdown of certain carbohydrates. Many organisms actually use carbohydrates as their primary energy source, so things like glucose and sugars and starches and things like that. And we find that glucose is probably the most common carbohydrate used in living cells. Glucose is then catabolized by a couple of processes, one of which is through cellular respiration, where glucose gets broken down into water and carbon dioxide over many steps. And it goes through the processes of glycolysis and the Krebs cycle and the electron transport chain. Another way that they can work is to go through fermentation, where glucose is also broken down, but you will end up with some organic waste products. And so if you're familiar with fermentation, you get things like alcohol at the end of it. So that's one of the organic waste products. Now let's look at glycolysis under the respiration set of reactions. Glycolysis um, is the splitting of our 6-carbon glucose into two pieces, and these are called pyruvates. So glycolysis breaks glucose into two pyruvates. And so you start with glucose. Oops, wrong thing. Glucose gets broken into pyruvate. How many? You get two pyruvates. And you're also going to make two ATPs. You're going to make two NADHs, which are electron carriers. And you're going to get those pyruvates that we mentioned. And these are going to be some of your precursor metabolites, sometimes called pyruvic acid as well. So the next thing you're going to see is pyruvate being broken into acetyl-CoA, and acetyl-CoA then going through the Krebs cycle. Now, since we've got two pyruvates, we'll break down to two acetyl-CoAs, and we'll go through the Krebs cycle for each of the acetyl-CoA. So we'll go through it twice. And I have a diagram for this one where you've got the acetyl-CoA produced. So pyruvate's a 3-carbon molecule. We make NADH as an energy source. We actually produce some carbon dioxide here too. We make acetyl-CoA. The next thing we do is we go around the Krebs cycle, and it produces NADH and ATP and FADH2 as we go around, and we do that reaction again. And so what that does is it's putting all this energy through this process into our NADH and FADH2 uh, electron carriers, which will take this reaction on to the next part of number 3 in our cellular respiration cycle. And that's the point where we're going to get lots and lots of energy, because we have made a lot of our electron carriers holding electrons, and we're going to dump those electrons and get a lot of energy out of them. So the electron transport, sometimes called the electron transport chain, is probably the most significant step of making ATP, because it's a series of redox reactions that allows for lots and lots and lots of ATP to be made. And so we end up with a series of our carrier molecules, like our NADH and FADH2s, that pass electrons from one, pass them off into this series of reactions, and they just keep passing the electron. That's why it's called the electron transport chain. It's like a bucket brigade, where one hands off the electron to another, and it hands it off to another, and hands it off to another, and hands it off to another. And so energy from this passing of electrons is basically moving the electrons through, and then as it moves along, it's able to pump protons across a membrane a lot of times, producing a proton gradient. And this is usually in our eukaryote versions of producing this kind of energy. So we don't see this inner membrane in bacteria, but we do see this in the mitochondria of our eukaryote organisms. And sometimes you'll be able to find it in there are certain cytoplasmic membranes inside the prokaryotes that will also be able to do cellular respiration this way, so they'll use a membrane. Now, the next thing that you'll see is the kind of a representation of the electron acceptor. As you can see, with each of these electron acceptors, you see NADH NAD plus grabbing an electron as it's been dropped off by some other electron acceptor, or picked up, I should say, back in one of the reactions. It then drops off the electron here, which moves it there, which pulls in a hydrogen, as you see, and it moves it here, causing FADH2 to drop off an electron, and we just kept seeing this being moved. And as we move along, dropping off these electrons, eventually it ends up with oxygen and hydrogens forming water. This is why oxygen is so important to our doing cellular respirations, where we have to have oxygen when we breathe. And it's not only us, but anything that's aerobic that uses oxygen to do energy needs oxygen to survive through this. And this is the reason, major reason for that oxygen. Now, as you see these electrons being passed along, we're using up the electrons from our NADHs and our FADH2s and our ATPs, and along the way we also get these hydrogens pumped in, and that's going to also create some ATP later after this cycle. So at the end of the electron transport chain, we see one NADH being oxidized to NAD+, which is going to produce 3 ATP. We see one FADH2 oxidized to FAD, which produces 2 ATP. And then in total, we see that one glucose molecule, through all the steps, would do this. Glycolysis producing NADH, there's 2 of them, and 2 ATP. The before Krebs cycle part, where we're producing acetyl-CoA, is going to produce 2 NADHs and 2 CO2s. This is where carbon dioxide is given off, like when we breathe out. Same thing for bacteria and other and the eukaryotes. The Krebs cycle will produce 4 carbon dioxides, 2 ATPs, 6 NADHs, and 2 FADH2s. Notice we're making a lot of these electron carriers, these NADHs and FADH2s. And then they go to the ETC, the electron transport chain, and they're going to make 34 ATPs. So in total, we're going to see 38 ATPs made from one glucose molecule, which is pretty cool. Without this, cells would not be able to produce enough energy to survive. Now, sometimes you're going to see this process listed as 36 ATP, and it's because the input to do glycolysis requires a little bit of ATP, and so usually it's around 2 ATP to go in. So it takes a little bit of ATP to make some ATP, but an investment of 2 for a product of 36 or a profit of 36 is good. And if you've ever heard the term, it takes money to make money, it's the same idea here. It takes ATP to make ATP. Now, there are times when you're not able to make as much ATP by using respiration, so you have to do something else. So when cells can't completely oxidize glucose by respiration through the Krebs cycle and electron transport chain, so if you don't have oxygen, they might do instead a different thing that's going to produce not as much energy, but some, called fermentation. And so we'll produce only 2 ATP and 2 NADHs. And basically, you don't get the Krebs cycle in the ETC. You will only do glycolysis. And then we'll have to have 2 NADHs that must be reoxidized. Basically, this is a little more wasteful process. It's going to end with some organic molecules being made instead of just a kind of a full breakdown where you get a bunch of ATP made and all the energy transferred to ATP. Here we can't do that, so we end up with some kind of organic waste molecules, which we call useful things by humans. We use some of them, some of that pyruvate or pyruvic acid that's made will go in and produce things that help us to make cheese or to make soy sauce or yogurts or to wine and beers, or even to acetone and isopropanol, alcohols that we use for cleaning. And so this is some products that we get from fermentation, but the product that the organism gets is survival and some ATP. Even though it's not a whole lot of ATP, they do get some to survive, hoping that in the end they'll be able to do some aerobic respiration with oxygen and produce more ATP later, if they're possible. Some organisms only can do fermentation and they are not able to switch back and forth, but some are. So some can do fermentation, some can do cellular respiration. It all just depends. So this just kind of gives you a little summary slide of respiration and fermentation side by side. What happens if you don't have what you need to do respiration? You'll end up going to fermentation. So it's a shorter process, it doesn't make as much. Respiration is a much longer process, but it does make a whole lot more ATP in the end.